So I'm just going to press go. So you've had a bit of time now to have a look at the course. How are you finding it? How are you feeling? How are you finding the level? Are you finding it difficult? Are you finding it easy? Are you managing okay with the written content? Can you let me know in the chat, please? It gives me a bit of an idea of where you're at with it. Probably have to go over it a couple of times. Yeah, that's understandable. Yeah, it's quite challenging, isn't it? It's a step up from school. So yeah, that's absolutely understandable. Bit nervous, nothing to be nervous about. You'll be fine. You'll be fine. Right, we're on session 2 today of the first unit. I'm just going to close some boxes down so I can see what I'm doing. So the first unit is cells, tissues and organs for healthcare. And we're on session 2 today. So in the last session, we had a look at the organelles, the cellular structures that make up the cell. So not just organelles, other structures as well. And we also had a look at calculations for microscopy as well. And we're going to move on today and have a little bit of a look more at the cells. So we're looking at this learning outcome. It's just a point really that we're looking at today, understanding the range of cellular specialisation in the body. So our cells are specialised. They're not all the same. They've all got the same DNA, but they've got different bits of that DNA switched on, which makes different cells do different things. So you have cells in your gut that are specialised for digesting and absorbing your food. You have nerve cells, which are specialised for sending nervous impulses, for example. So we're going to have a look at some of the cell types. There's 4 different main types that we're going to consider. And these give rise to 4 different tissue types that we'll look at as well over the next couple of sessions. So we're going to explain how the structures of the 4 human cell types are linked to their specialized functions. So we're going to have a look at some specialized cells, and we're going to have a look at 4 different tissue types as well, which are basically made up of these specialized cells. And then, depending on how we get on for time, we might move on to have a look at stem cells as well. We'll see how we get on. So explain how the structures of the 4 human cell types are linked to this specialized function. That's what we're up to today. Let me just move another box. It's the thing with Zoom, you have boxes everywhere and they get in the way quite often of your slides. So let me move that right out of the way. And another one to move out of the way. Okay, so we had a look at the cell last session, and we had a look at the structures that make up the inside of the cell and the membrane, which is really important on the outside of the cell. And these structures making up this cell make up the smallest structural and functional unit that is classified as living. So individual cells can be classed as living cells, and they can live actually independently. They can live, they don't have to be in an organism. So we can take human cells and we can grow them in the lab. They don't have to be part of a tissue or a organ or a human for that matter. We can grow them in fluids in the lab, and we do as scientists. We do that so that we can study them. We can see how they respond to drugs, that we can see how they respond to various treatments before we would test them in animals and humans, for example. So those cells that we grow in the lab, we would classify them as living. They are living cells. So cells are the smallest structural and functional unit of living things, the smallest thing that we can say is alive. So if you get smaller than a cell, it's not living. And that's why viruses are often not classified as living, because they're not made out of cells. They are complex molecules, yes, but they are not cells. So most people would not describe viruses as living. So you have to be either one cell big or multiple cells to be classed as living. So bacterium, for example, is one cell big. Some fungi are one cell big, like yeast is one cell big. So they're called single-celled organisms. There's quite a lot of them. Loads of them cause disease, and we'll meet them in a later unit on disease. But in humans, we are multicellular organisms, and our cells are arranged into groups, and we call these groups of cells tissues. So these tissues are made up of cells from the same origin. So they're really similar, not necessarily all identical, but they're very similar to make a tissue. And then that tissue will carry out a specific function. So each tissue has got its own job. And that's why the cells have to be specialized, because the tissues have got to do a certain job. So they need specialist cells for those jobs. And then from the tissues, we have organs. So we're all familiar with the idea of what an organ is, I guess. I'm assuming. So like your stomach is an organ, your brain, your heart, your pancreas, your liver. These are all organs. And what these are groups of tissues that perform a specific function or group of functions. And that could be different tissue types. So for example, your heart is made up of muscle. It's cardiac muscle, but there are other tissue types that are making up the organ as a whole. And we'll have a look at that, I think in maybe the next session. Maybe it's today, I can't remember which lecture it's in, but we'll have a look at that soon, to have a look at the different tissues that make up that specific organ. The stomach is another good example. It's got some tissue called epithelial tissue, which we're going to talk about a bit today, but it's also very strong muscular sac, which is how it can churn up your food. So that's two different tissue types there already, the epithelial and the muscular tissue. So the organs are groups of tissues, and they're all working together to form this one specific function. So cells are often specialized to carry out a particular function. That means that they have one particular job to do. And because of this, they can have a very distinct shape and structure. They might have special kinds of chemical change that take place in their cytoplasm as well. And these changes in shape and potential chemical reactions that are different in each individual cell enables them to carry out its specialist function. So the cells start off the same when we are in a sort of embryo form. All of our cells are identical, and they specialize as we develop in utero, in the uterus. And this happens by different genes being switched on in different cells. So all of the cells, as we'll see fairly soon, actually have the potential in an embryo early on, have the potential to become any cell. But different genes are switched on in different cells, so they specialize. So if it's going to become a nerve cell, the genes that make that cell into a nerve cell will be switched on, and they will specialize and grow into a nerve cell. And if it's going to specialize into a heart cell, cardiac muscle cell, it will specialize because the genes to make it into that type of cell type will switch on. And when we're talking and describing these specialized cells, we say they are adapted to their function, and we call these tissues then. So the first one we're going to have a look at is the epithelial tissue. So epithelial tissue is made up of epithelial cells. And this tissue appears usually as large sheets of cells, so like flat and covering surfaces. Think of it a bit like the skin, but it's on the inside. So it covers all of the internal cavities. You can see it in your mouth, for example. It's that type of tissue. So it's not quite skin, but it covers large areas like the skin. And what it does internally is it helps to keep your organs separate from each other, because they can and they do actually stick together in, I mean, obviously, like that's not something that should happen, but it can and does happen in people, and when they're not well, your organs can stick together. So these epithelial tissues help keep the bodies separate, and they help to keep them in position as well, and also provide some level of protection for them. So epithelium is the singular word. Epithelia is plural. And epithelium is found in lots of different shapes and sizes, and that variability in the shapes and sizes of the cells that make up this tissue is a sort of specialization of the cells of this tissue type. And we're going to have a look at some specialists examples of these cell types as well. So on the board at the minute, we've got something called squamous epithelium. Also, I've heard it pronounced squamous as well, so I'm not actually 100% sure which is the correct pronunciation. I think squamous, to be honest. Because it looks like a square. You can see it's from the same word origin as square. So it's basically square-like cells, and they're found as a flattened layer. So they're flat, like a square, and they're a bit like tiles that you see on the floor or on bathroom walls. You know, they're just square shaped, covering a large area. And this is what we've got inside our inner cheek. And actually, if you go and have to give a swab because you've been at a scene of a crime or something, and the police will go in to take a swab, that's what they're scraping when they put those little cotton buds in your cheek. They're scraping the epithelium cells off and getting some DNA out of those cells. Let's get this page turned over. So other epithelial cells are specialized, and they contain microscopic hair-like protections called cilia. We mentioned cilia yesterday, and I said we'll talk about it today, so we'll come on to this today. These cilia are the projections, they're bits that stick out. So instead of it being sort of flat, let me just make sure you can see me on the screen, because I can't quite see it. So yeah, there's my hand there is like a sort of, rather than it being a sort of round surface, there's bits that stick out, and they tend to wave the cilia or in lots of tissues to move things along over the surface of the cell. So that's quite an important function, that cilia. But also they might just have these cilia projecting and increasing the surface area. You know, there's a huge surface area there with all of my fingers compared to if the cell was just round like that. So it's a way of increasing the surface area as well. So if we have epithelial tissue like this, or epithelial cells that have got cilia on it, we call it ciliated epithelium. It's actually found, we have some in the gut, we have some in the female reproductive tract, and we have them in the respiratory tract as well. So the upper respiratory tract. Now this is a bit disgusting, so apologies if you've just eaten dinner, but you may have noticed that you get mucus in your upper respiratory tract, and that then gets moved to the back of your throat and you swallow it. And that's because the ciliated epithelium move this mucus away so it doesn't go into your lungs. You actually swallow it and it goes down into your stomach, where it's broken down by the acid that's in your stomach. And also, which is even more disgusting, is it links up to your ear, and the fluids, excess fluids from your ear, also end up going down into your stomach as well. So that's what it looks like. So you can imagine these cilia waving like this on the top, and it will wave that mucus to the back of your throat where you swallow it. And instead of going into your lungs, because if that went into your lungs, it would be a lovely environment for loads of bacteria to grow and infection to develop. So instead of all that mucus being in your lungs, it goes down into your stomach and gets broken up by the acid there. It's got to be there though. It's not like, well, why have we got it if we just want to get rid of it into our stomach? It's got to be there because it traps dust and pollen and dirt and bacteria and things like that that we breathe in. So we can classify epithelia based on 3 criteria. We look at the number of layers. So it's called simple if it's got a single layer. So both of these are simple epithelia because they've got 1 layer of cells. And if it's got more than 1 layer, like 2 or 3 stacked on top of each other, we say it's stratified. We also categorize it or classify it based on the shape of the cells. So here we've got squamous epithelia, and the outermost layer is flattened. It looks like tiles or scales on a fish. And we've also got columnar epithelia. That's when they're tall. And you can have cuboidal epithelia, which looks like cubes. You know, no prizes for being inventive with the names here. So the specializations on these cells on the surface, such as cilia, allow them to perform their specific function, like a barrier to infection here. So we're going to have a look at a specific example here. These are goblet cells, and they secrete mucins, which create a protective mucus layer. What mucins are is highly glycosylated proteins. So that means they are proteins that have got carbohydrates attached. So it's just a normal protein, but with stuff stuck to it. Remember last session, we talked about proteins quite a bit because we were talking about all these structures inside the cell having these important roles. And we said, you remember the nucleus contains the code to build the protein. The endoplasmic reticulum is where the protein is made, the ribosomes, and then the Golgi is where the proteins are modified. That Golgi apparatus, that lava lamp-like structure, is where the proteins are packaged. So that's what's going on in the Golgi there. We're sticking on these carbohydrate groups to make our proteins specialized and glycosylated. That's what's going on in that Golgi apparatus. So these particular cells, these goblet cells that we're looking at in this example, this example of specialized epithelial cell, these are found in the trachea and the intestines. So the ones I was just talking about that move the mucus along. Hugely important in terms of maintaining the integrity of your intestines, of your respiratory tract, because the respiratory tract is extremely vulnerable to infection because it's what we call an exchange surface. So that means we're exchanging materials with the outside environment. So we're exchanging oxygen and carbon dioxide through our respiratory tract. So it's really big surface area, which is really important for exchange, but it's vulnerable because it's in contact with all the dirt, all the pollution, all the dust, all the pathogens from the outside world. So this is a first line of defense from infection. And they're also quite important in the stomach. So in the stomach, we have stomach lining. You can see this bottom layer here. And we have a mucus layer over the top. And we've got these things called gastric pits, and they secrete some really nasty stuff, hydrochloric acid, which is very acidic. It's got very low pH, so it's very, very strong acid. And they also secrete pepsin, which is an enzyme which breaks down protein, which is great because we eat loads of protein. We want to break it down, but also this stomach lining is made out of protein. So that's not so great because these enzymes could damage it. So the production of mucus is really important in this area as well. So this is a goblet cell then. This is what they look like. It's a specialised epithelial cell. And they get their name from looking like a goblet. Now, you might look at that and think, that doesn't look like a goblet to me, and it doesn't really look like a goblet to me. But if you look down here, you can see that sort of almost goblet-like structure. And it's what they look like on the microscope slide. So it's not what they look like in real life, what we now know they look like. It's what they would have looked like when people were looking at them first, that they get these crazy names from. So when people first looked at this tissue, they'd have been seeing slides like this. And you can see these sort of goblet-like structures down there. So that's how they get these weird names. So they are about a quarter wide as they are tall. So what that means is its width is about a quarter of its length in size. So you could fit 4 of its widths down its length. And they are polarized. So what that means, that's a fancy way of saying they're not very balanced out in where this stuff is. So it's asymmetric. It's all bunched down one end. So the nucleus and the other organelles are located at the basal end. That's the bottom end. So they're all down here. So we can see the Golgi here. We can see the endoplasmic reticulum that we talked about last time. And we can see the nucleus here. And what do you think this structure is here? Anyone want to have a guess in the chat what these little pink things are? Anyone remember? Want to have a guess what organelle that is, what cell structure? Mitochondria. Good work. Well done. Excellent. Yeah, so these are little mitochondria. You can tell they're mitochondria because they're sort of sausage shape, but they're not long sausages. They're quite fat sausages, mitochondria. And you can just about make out that double membrane that we talked about as well there. Okay. So at the top here, we've got these secretory vesicles, which contain mucin. So this is a really good example of a specialized cell. So we've got the nucleus here that will contain DNA. And when the mucin producing genes are switched on, the DNA will be copied into a messenger molecule that will go to the endoplasmic reticulum, which has got the ribosomes on. Remember, the ribosomes are the site of protein synthesis. So it's going to make this protein in the ribosome. Then the ribosome is going to send the protein off to the Golgi apparatus. That protein needs to be highly glycosylated. It said on the last slide, highly glycosylated. That means it needs a load of sugar stuck to it. So it's going to go into the Golgi, have a load of sugar stuck to it, sugar molecules, carbohydrate molecules. And then it will be packaged into these vesicles, which will then move to the apical end of the cell, apex, top, basal, bottom. It's going to move to the apical end and release these secretory vesicles into the gut here. So a feature of these goblet cells is extensive endoplasmic reticulum and Golgi apparatus in the cytoplasm. And based on what I was just saying, there's a load of mucins released from here, which are proteins. That makes sense, doesn't it? Because they've got a lot of the machinery which is needed for protein making and processing. So the endoplasmic reticulum, it's got all these ribosomes on, these ribosomes for making proteins, and the Golgi are there for processing the proteins. Do those vesicles go out of the cell with the mucins as well? They do. Yeah, that's exactly what happens. Good question and well identified. So that's where the Golgi is packaging the mucins, and it's packaging them into these little vesicles, these secretory vesicles, and then they'll just move up to the top here. They don't come out of the sides, because if you look at the structure here, they're packed together, one against each other. So they can't really release stuff out of their sides because it's just going to hit the cell next to it. So they need to get these secretory vesicles to the top. All of these mucins are packed in, and then they can be released out into the gut in this case. So this Golgi apparatus, this structure here that we talked about packaging these proteins that add in these carbohydrate groups. Carbohydrates are, that's what the glycosylating is. It's adding sugar groups or carbohydrate groups to the protein. They're added by enzymes called glycosyl transferases. So transferase just tells you it's transferring something. Anything that ends in the A's name is you pretty much guaranteed it's an enzyme. So we're going to learn about salivary amylase. You might remember that from GCSE. We're going to learn about proteases. We did mention earlier that these what were we mentioning? Something about proteases. Oh yeah, I was talking about proteases being in the gut, wasn't I? So protease means it digests protein. Salivary amylase, you might just remember from GCSE. So anything ending in A's is an enzyme. And because it's a transferase, that means A's tells me it's an enzyme. Transferase tells me it's an enzyme that transfers something. And because it says glycosyl transferase, that just tells me that it's an enzyme that transfers a glycosylate group or something onto the onto a protein. So the mucins are then put into these secretory granules or vesicles, as I tend to call them, but it's the same thing. And then they're transported up to the membrane. They fuse with the membrane through exocytosis. And we talked about that last session. We talked about the fact that the membrane acts very much like a lava lamp, where bits can pinch off and re-emerge. So that's going to fuse with the membrane and then pinch off and be released into the lumen of the organ. So lumen's a bit of a funny word. It just means hole in a tube. So if you think about a toilet roll tube, the hole through the middle of it would be called the lumen. And we talk about the lumen of the gut because your gut's a tube. So you've got your small intestine and your large intestine. We'll say it's got a lumen because it's got a hole through it. Your arteries and veins, they're tubes. So we say they've got a lumen as well. So that's all that means. It just means the hole through the tube. So because these secretory vesicles have got to be released up there, that's what you would see nearest the top of these specialized cells, towards the edge of this epithelial tissue. And we call that the apical end. And apex is, you're used to that sort of structure being, that word being used for apex, like the apex of a roof, aren't you? So that's what apical end means. It means the top end of the goblet cell. And then if we look here, you can see there's microvilli, and those are those sort of little structures. You can see them up here. They look like fingers. You can see they're not hairs, but people often describe them as being hair-like because they look like hairs under the microscope, but they're much more like fingers if you look at it in this diagram. And they increase the surface area because if you think that little finger wasn't there, you just have this tiny bit of surface area. But by having this big finger-like projection, you're increasing that surface area maybe like 8 times just from that one microvilli structure. It's just basically a way to maximize the space available for secretion of the contents into the lumen. Okay, let's have a little go at some questions. I'll leave that up. You can write in the text chat if you want. You don't have to if you're not feeling brave enough, but it's great if you can because it lets me see how you're getting on. But if not, do have a go and write it down in rough paper, so you can track how you're getting on. So why are the goblet cells named as such is number one. And where are the nucleus and most organelles located in a goblet cell? And what is the function of the microvilli at the apical end of the goblet cells? The apex, the top end. Okay, good work. I can see a few of you having a go. Well done. I'll give you a minute. Let's have a look, shall we? So, goblet cells are named as goblet cells because they look like a goblet, or perhaps not in real life, but at least on the slides of the tissue. So where are the nucleus and organelles located in the goblet cell? They are located at the basal end. So they're squished more towards the bottom. We've got the secretory vesicles at the top and let's have a look, and the organelles towards the bottom. Okay, so here's the organelles or the cell structures, if you prefer. They're all sort of down the bottom end. And then we've got these secretory vesicles at the top. Not many of the actual brain-bound organelles that do jobs like making. And what is the function of the microvilli at the apical end, so the top ends, those little fingers that we saw? They're there to increase the surface area to secrete those mucins out of the cell. Another tissue type now. Things. Okay, so connective tissue cells. Connective tissue is the matrix that we find underneath this epithelial layer. And this layer is important in connecting or supporting most of the organs. As an example of connective tissue, it's generally sparse. It hasn't got a lot of cells in it, but it contains what we call extracellular matrix. So extracellular just means outside the cell. So there's a lot of tricks. It just means stuff. So there's a lot of stuff out by an extensive extracellular matrix. So blood has got an extensive, has got a lot of stuff outside the cells. And this is could be glycoproteins. So again, proteins with sugars or carbohydrates attached, and proteoglycans as well, another type of molecule. And what this connective tissue does is it provides structural and mechanical support to other tissues. So blood is an example of connective tissue. Bone is an example of connective tissue. And it can bring about exchange of nutrients and waste between the circulation and other tissues. So it's got some sort of exchange roles, the blood, for example, and it can help with the repair and regeneration of other tissues if they are damaged. So the two main components are the extracellular matrix, so the stuff that's outside the cell, which is mostly proteins and fibres, and support cells. So the matrix itself, so the stuff that's outside of the cell, basically consists of fibres embedded in some sort of support medium. And that support medium could be a solid support medium, a liquid, as with blood, or a gel. There's lots of different types. We've got loose connective tissue, cartilage, fibrous connective tissue, adipose tissue, which is like fat, basically, blood and bone. There's 3 different types of fibre that we're concerned with. So collagenous, elastic, and reticular there. And see if you can answer these couple of questions for me. What is the connective tissue from epithelial tissue? And what are the following is not a function of connective tissue? See if you can give me the answer for those. Oops, let me open the chat. That's what I'm trying to do. Okay, good work. We'll have a look in a second. There we go. So answers are B and C. Connective tissue is sparsely populated with cells. That means it hasn't got much in terms of cells, but it's got an extensive extracellular matrix. So yes, there's cells, but there's a lot of other stuff as well. We're going to have a look at blood later on in the course, but blood's a really good example of it, something being sparsely populated with cells. Yes, there's blood cells, there's red blood cells, there's white blood cells, but a lot of blood cells, it is the plasma fluid. That's why it's a liquid. Plasma by volume of blood. So most of it isn't blood cells. Most of it is this extensive extracellular matrix stuff outside the cells. So what's a function of connective, which is not a function of connective tissue? It doesn't secrete enzymes for food digestion. That happens inside the digestive tract, but it does provide structural and mechanical support for other tissues. Think about bones, for example. It does facilitate exchange of nutrients and waste between tissues. That's blood, for example. And it does assist in regeneration and repair of damaged tissues. So blood has got a role in that as well. So let's have a look at our examples here that we were just talking about. So blood is an example of a connective tissue. Its matrix, so remember that extensive extracellular matrix, the stuff outside the cell, is a liquid called plasma. That liquid you will have seen when you've cut yourself, particularly as a child, if you got a graze on your knee, you know, when you sort of skid across the floor and you take a layer of skin off and it goes all sorts sticky and wet as the wound is healing, and you get this sort of seeping of this yellowy liquid. That's the plasma. That's the extracellular matrix that we find. And we did talk about there being fibres in this extracellular matrix. You don't read, but they become insoluble during clotting, or scabs start to form. And the main function of the blood as is transport of nutrients and hormones and things, but also defense against infection. So the other connective tissue we were talking about as an example is bone. So the matrix type in bone is solid or semi-solid. And that's how you get such a sort of, there are fibres present in the matrix, and the main function of this binding, protection, as well as other things like making blood cells, for example. Attachment site muscles is another example. So bone, then, the matrix is hard or calcified. Cells making up the bone have got special names: osteoblasts, osteocytes, osteoclasts, and its main function is support, protection, movement. And other connective tissues, like the blood example, have this soft or fluid matrix. They have different types of cells, like red blood cells, white blood cells in the example of blood, different types of fibres, and different functions like binding, transport, and flexibility. Okay, we're going to have a look at a specialist connective tissue cell now. And this is a specialised blood cell. So this is a macrophage or macrophage, some people pronounce them as. So the macrophages are found in connective tissue, blood. They're actually part of the immune system, and they go around protecting the body from infection. So we talked about the epithelial barrier earlier. We talked about this layer that we get inside our mouth. We talked about the lung being really important with these ciliated cells to keep bacteria, viruses, etc., out of our lung tissue. But if a microorganism does cross this epithelial barrier and somehow begins to replicate in the tissues of the host, it will be immediately recognized by the phagocytes and macrophages that live in these tissues. Well, live, I suppose, they patrol would be a better term. So macrophages can mature continuously from circulating monocytes. So what does that mean? Well, circulating monocytes are white blood cells that are just circulating in the blood, in the tissues. They leave circulation to migrate into the tissues throughout the body. So they're essentially like on patrol through our tissues, looking to find stuff that, and if they find stuff that isn't part of our body, they will mount an immune defense. So that might be, in this case, we're talking about a microorganism that somehow found its way through the barrier, the epithelial layer. So what would happen then is a macrophage would do something about it. But it doesn't have to be that it's a splinter that's pierced your skin. It could be that you've inhaled some pollen. So anything that shouldn't be there will get noticed, and these macrophages will then do something about this invading pathogen. So they've found in, especially like blood and in connective tissue that is associated with, for example, so that's the digestive system and the lung and throughout the spleen. And what they do in the spleen is they remove senescent blood cells. That means old blood cells that are just too old. They need to remove them. But in the gastrointestinal tract and the lung, these are exchange surfaces. We're exchanging stuff with the environment. As I said earlier, the lung, we're breathing in oxygen, but also bits of pollen, bits of pollution, bits of bacteria. The gastrointestinal tract, we're taking in food, but also whatever is on our dirty little fingers. If we're eating with our hands, we're taking in bacteria and all sorts of horrible stuff that needs to be dealt with as well. So these macrophages are found in large numbers where they're really needed at these exchange surfaces. They're needed to protect us, and also at the spleen where they're going to be needed to break down and remove these old red blood cells. So the macrophages are indistinguishable from something called fibroblasts, but they can be by using special dyes and carbon particles and stuff when they're being stained in that they're different from these cells, fibroblasts, which are, I haven't really talked about, but there's a lot about them on the course. So what makes these macrophages interesting to us is they do something called phagocytose foreign material in the connective tissue layer. So they undergo this process called phagocytosis and play an important role as antigen presenting cells. So they will label up, they use antigens like a little flag. It's like a red flag saying, right, there's a problem with this. And they can use antibodies to attract other macrophages and phagocytes to the area. So we're going to have a look at this process of phagocytosis now. So during infection, the immune system will begin to mount an attack, as we saw with the macrophage. There's three things they can do broadly. There's lots of things. It's very complex. This is a very brief summary. They can undergo phagocytosis. They can phagocytose and engulf the pathogens. They can produce antibodies, which act as a little flag to notify the rest of the immune system there's a problem here, come and deal with this problem. And they produce something called antitoxins as well, which tries to stop the damage of any toxins that's been released by these invading cells. So phagocytosis then, we're going to have a look at what's going on here. So this is a macrophage. So macrophages are phagocytes. So phagocytes are cells that can engulf and break down by phagocytosis. So macrophages can do this process, phagocytosis. They can phagocytose these bugs, bacteria that have been inhaled or eaten accidentally. So what this macrophage is going to do is it's going to put these little like arms almost around these bacteria here. Then it's going to close off that and bring them inside the cell where they will then get broken down and digested. So it kind of loops around and hooks them in, brings them in and breaks them down. And that process is called phagocytosis. So the phagocyte engulfs the foreign body. We say foreign body because it's not necessarily a bacteria. It could just be virus, pollen, dust, anything that shouldn't be there. And it destroys it by breaking it down with special enzymes once it is inside that macrophage cell. So really, really important defensive role there. So macrophages are specially adapted for their function through their plasticity. What do we mean by that? They really well look at the shape of it here. And that's not a round cell, is it? It's plasticity means it can change shape. So it can move around, it can change shape, and it can engulf these bacteria. So they change their behavior based on their environment, and they have this really irregular shape, which actually helps them to squeeze through the capillaries. So the capillaries are very, very thin blood vessels that get right into our tissues. And these macrophages can squeeze through the walls of the capillary because of this plasticity. And we call this these sort of arms that go around the bacteria, we call these pseudopods. That actually means, pseudo kind of means like fake. Like pseudonym is a fake name. So pseudopods means fake legs. So it's got these, they look more like, they're more like arms in the way they sort of engulf and bring the bacteria in. But it has these fake legs, fake feet, I suppose, not legs, that engulf these, whatever they are, bacteria. They have specialized organelles like lysozymes, which are there to digest and break down the stuff that is engulfed and brought into the cell. And they have a really large number of receptors to detect pathogens, and they can alter their gene expression to switch between two states, either inflammatory or tissue repair states. So that's quite an important function there. They're also equipped with the machinery for something called the respiratory burst, which is reactive oxygen species production, ROS production, and that will kill off any pathogens that they have engulfed this pathway. So they can basically kill off the bacteria using this special reactive oxygen species production, which is a load of molecules which cause damage. Okay, what is one major role of macrophages in connective tissue? Then have a look at this and see if you can work out what the answer is there. Good, I can see some people writing in the chats. If you're not sure, make sure you do have a go. If you don't feel confident enough to write on the screen, that's fine, but have a go at home. Okay, so there's the answer. The specialist role is to phagocytose the foreign materials. So foreign materials just means anything that isn't part of the body, anything that shouldn't be there. Let's have a look what that just said. A quick question. I have a question. When a pathogen, when someone gets in, what is happening or failing with the macrophages for them to get infected anyway? Oh, it's really complicated and really complex. The macrophages are initially they're non-specific. So they just go around looking for stuff that shouldn't be there. And it takes a few days for the immune response to get going to be very specific to the infection. So if you're infected with something that manages to multiply really quickly, quicker than your immune response can get mounted, because these are non-specific, they'll just go, oh, that shouldn't be here, that shouldn't be here, that shouldn't be there. You know, it could be a bit of pollen, it could be a bacterium, it could be anything to start with. You need to mount a very specific immune defense response with specialist cells that are just looking for that particular pathogen for it to work effectively. And yeah, so it's a bit of a, I think we will be getting onto that when we do the disease unit though, but if that's not that might be a specialist unit. I'm not sure if that's one you have to pick. But yeah, we will be having a look at that eventually. And here is the last type of, another type actually of specialist cell. These are muscle tissues and they're responsible for movement, obviously. Muscle cells are what we say are excitable. That means they can respond to a stimulus. They're connected to the nerve cells. There's something called a neuromuscular junction that connects them. They're contractile, so that means they can contract. So if you think about your arm like this, when your bicep contracts, it brings about a movement with a pulling force. It brings about the movement of your forearm, bringing those two bones together. So we've got lots of different types of muscle. We've got skeletal, which I was just showing you with the bicep movement. We've got cardiac that we talked about briefly before. And we've also got the smooth muscle, which is found inside your organs. And we'll talk about that when we do, well, we'll talk about all of these because we're going to do muscles, we're going to do the heart, and we're going to do the digestive system, which has got a lot of smooth muscle in it. So we'll talk about that a lot later on. But we're going to have a quick look at the muscle cells now, which are adapted for their function. They're striated. We say striated because they look stripy under the microscope. They are multinucleated. That means they've got several nuclei because they're made up from lots of cells merged together in their sort of early development stage. striation, or the stripes, is due to the alternation of these proteins, actin and myosin. We were talking about fibres and microfibrils last session. So that's where what these are, these proteins, actin and myosin. And what they do is, you can see here, they're sort of layered like this, and then they get pulled together, and that's how the muscle contracts and lengthens as they get pulled into each other like this. So these proteins are called contractile proteins. There's a lot of labeling of these bands, because you can imagine when they first got discovered and looked at under the microscope, that people were just seeing this. They couldn't zoom in any further before electron microscopes, and they could just see all these different bands. So all of these different lines that you can see on the image have got names. They're not really that important. What is important is the fact that we've got these two different proteins, actin and myosin, working like this. The fact that we get these sort of bands that we can see isn't that massively important, to be honest. So do you want to have a quick go on this one? What is the which of the following type of muscle with its location, which is the correct one? You'll see, and which causes the striated appearance on the muscles. Fab. So the correct answer there is cardiac muscle, which makes up the heart wall, and that can contract involuntary, because obviously if we had to think about our heart contracting, we'd die in our sleep, because we wouldn't be able to think about it. And the alternate arrangement of those two types of filaments, those fibers, those muscles, actin and myosin, give it the stripy appearance there. The muscle cells, not that it's particularly important now, but we'll talk about them later. They're very similar, you know, they've got the same standard structure. They have membrane, cytoplasmic, endoplasmic reticulum, but we, unfortunately, to make life worse for ourselves and more difficult, give them specialist names in muscle cells. We call them, instead of the plasma membrane, we call it the sarcolemma. Instead of the cytoplasm, we call it the sarcoplasm. And instead of the endoplasmic reticulum, we call it the sarcoplasmic reticulum. So that's something to look forward to when we learn about muscles. They've all got these many nuclei and a large number of myofibrils or fibrils arranged so that they can contract when the muscle needs to be made shorter. Is there a reason they're given different names? Do you know what? I don't know. It's probably something historical. It often is. They probably started looking at it as tissue instead of individual cells and named it before they realized, or something along those lines. It usually is. Is that a change in names for the muscle cells only? Yeah, just for the muscle cells. So we've got these, the important thing about the muscle cells is these alternating bands, alternating fibers. These bands have got different names, because they're not particularly important, to be honest. It's just what makes them stripy. But we've got the I band, the Z line, the A band. They've all got specific names, and the M band. You can tell it was a lot of German scientists that were working on this by the names. I think German, maybe Austrian, something like that, Swiss maybe. But they've all got very Germanic sounding names, these lines. So the really important thing there is the actin and the myosin, and the fact that the individual fibers don't change length during contraction. It's just that they are drawn together. That's what does that. the contraction. Okay, so I think where we're up to now is we have done enough to answer some of your bits of your assessment, which you'll be learning about in tutorial. But you should be able to explain the microstructures in the diagram that you'll be given. You should be able to do the magnification calculations, and you should be able to not do this stem cell stuff, but identify different tissue types and briefly describe in the body. And then next session on Saturday, or whenever you watch the recording, we will have a look at this stem cell part of the assessment. So if anybody got any questions, now's a good time. If not, I think we're almost out of time. How are we doing? Yeah, just 1 minute over. So if you've got any questions, let me know. If not, off you go. Have a good evening. And I'll see you Saturday. Bye, guys. Bye. Bye-bye.